Thank you. Good afternoon. Thank you for having me here. I know Dr. Shah says 10 years ago we may have a vaccine, but I think we'll understand more, and I think we're one step closer to that. Okay. So, as everybody knows, the atherosclerosis is currently being viewed as an immune-mediated chronic inflammatory disease of arterial walls. And adaptive immunity plays a very important role in this pathogenesis. As you can see, the adaptive immune response started with antigen-presenting cells taking up antigen, processing and presenting down the MUC molecules, and talking to naive T cells, and then differentiate T cell into pro-inflammatory T cells such as Th1, Th17, and NKT cells. And these cells, along with their inflammatory cytokines, interferon gamma, TNF alpha, IL-17, can interact with the arterial wall and make the wall vulnerable to atherosclerosis formation, especially in the presence of hypercholesterolemia. As everything in the world is also a good side and the bad side. I just talked about the bad side, and the good side is the immune response can also has a good counterpart. Under certain conditions, the antigen-presenting cells can take out the antigen and become tolerogenic. And these tolerogenic cells will talk to naive T cells and differentiate them into regulatory T cells. And these cells has a marker with CD25, FOXP3, and they typically secrete IL-10 or TGF-beta. And the cells and the cytokines can counteract the pro-inflammatory immune response, therefore achieving arthroprotective effect. And we had known for a long time the antigen involving atherosclerosis, LDL, or its oxidized form, Hisha protein, beta-2-glycoprotein 1. And this picture shows the colocalization of LDL and the macrophage. Now the question is, are they the good guy or are they the bad guy in the plaque? So we asked the question about 10 years ago, will immunization with LDL or LDL worsen or reduce atherosclerosis? Initially we thought it should worsen because all the bad guys come together. But our data prove us wrong. So when we immunize the cholesterol-fat rabbit with LDL, and we found there's a significant reduction of aortic atherosclerosis, and when compared to the non-immunized group, even in the presence of very high cholesterol level in the range of 1,000 milligrams per deciliter. And later on, similar findings are being reported by many groups in the world using mouse model of hypercholesterolemic-induced atherosclerosis. These are just a few examples. So far, it confirms the finding that if we immunize animal with LDL, that can reduce atherosclerosis. But the problem is LDL is a complicated molecule, and if we're going to use it as antigen, we have to isolate it from human blood, and this isolation process will have some safety concern. And also, since it's a heterogeneous big molecule, it's very hard to identify the precise antigen entity. So our lab, in collaboration with Dr. Jan Nelson lab in Sweden, so we start to look at the protein component of the LDL, which is ApoB100. Here's the protein sequence. To keep the story short, we basically established a library of 302 peptides and did a very rigorous, long screening process, and to identify the peptides that have immune response. And we also test those peptides and screen them as antigen for vaccine formulation. We finally filtered down to one peptide. We call the P210. This is the sequence. It's located in the amino acid position 3136 to 3155. And we tested it in the mouse model. As you can see from the bar graph here, the mice which immunized with the P210 developed significantly reduced atherosclerosis in the aorta compared to the non-immunized group and the adjuvant group. And this is the picture of, represented picture of the aorta from each group. And we also observed reduced macrophage infiltration in the plaque in the immunized animal. We also observed reduced dendritic cells, which is antigen-producing cells in the plaque in the P210 immunized animals. And to further understanding which cell components that mediate this effect, so and one experiment we did, so we transferred the CD8 T cells from P210 immunized donor mice into non-immunized recipient mice. As you can see, the recipient mice, the mice received P210 CD8 developed reduced atherosclerosis compared to recipient received CD8 from control groups. So far and if we transfer B cells, there's no effect. If we transfer regular T cells, CD4, CD25, there's no effect. So far, we believe CD8 T cell is the mediator of our vaccine. And also many other groups report the use of different delivery method to deliver P210, and either by dendritic cell approach or by a fusion protein approach delivered through intranasal, or just simple prolonged subcutaneous infusion of P210. They all reported reduction of atherosclerosis. Therefore, it seems to me P210 is a very promising antigen that can be used as in the vaccine. Now I have to change our focus a little bit to a different vascular disease, which is abdominal aortic aneurysm. And in a mouse model, if we infuse angiotensin to mouse, the aorta will become aneurysmal. And this picture just shows different degree and various form of aneurysm in the aorta. And type 4 on the far right is the most severe form and prone to rupture and cause death in the animal. And the picture on the left shows how we identify the aneurysmal segments in the aorta. We basically look at the 8 different segments of the whole aorta, then count the segment that become aneurysmal. As you can see on the bar graph on the right, the P210 immunized animal has the least amount of aneurysmal segment in the animal. And more importantly, this also reduced the incidence of rupture and increased the survival of the animal. As you can see, P210 immunized animal has the highest survival rate. So the mortality only about 6%. And as I mentioned in the first few slides, Th17 cells are very important pro-inflammatory cells causing response for the disease. Therefore, we look at the Th17 cells population in the immunized animal. As you can see, this pro-inflammatory subset of CD4 cell is significantly reduced in the P210 immunized the animal. So we believe this could be one of the mechanisms that are responsible for the reduction of aneurysm. As everybody knows, angiotensin can induce hypertension. And this is to our surprise. When we measure blood pressure on those animals, as you can see, the hypertensive response to angiotensin infusion in P210 immunized animal is greatly reduced compared to the other 2 groups. This is the picture shows when we measure whether it's a systolic blood pressure or mean blood pressure. And angiotensin 2, as you know, it acts on the angiotensin receptor. So next step, we trying to see whether this immunization changed receptor expression in aorta. So we took out the aorta and did the Western blot analysis. As you can see, the P210 immunized animal has the least amount of angiotensin 2 receptor expression. And therefore, we believe this downregulation of angiotensin II receptor in aorta could be responsible for the antihypertensive effect. So in conclusion, immunization with selected ApoB100 peptide reduced atherosclerosis and favorably modified plaque phenotype in hypercholesterolemia mice. Immunization with selected ApoB100 peptide also reduced angiotensin II, induced hypertension, aneurysm formation, and the mortality due to rupture. And we believe this observation provides proof of concept regarding the feasibility and efficacy of ApoB100 peptide-based vaccine for vascular diseases. And I'd like to thank my group at Cedars-Sinai Medical Center and Dr. Janssen Lab, and also the support from CardioVax and the various funding source. Thank you very much.